This video demonstrates a procedure to conduct differential imaging of two Raman resonances associated with biological structures with coherent Raman scattering signals. First, three synchronized short pulse lasers are overlapped in time and space. The overlapped pulses are then sent to a standard inverted microscope, which allows for the generation of coherent Raman signals from microscopic objects. Signals are converted into images using commercial software in order to compare different Raman signals that are produced. Results indicate that the difference between images obtained from doubly and singly resonant Raman signals provide enhanced sensitivity for weak Raman signals.
Hi, I'm Tyler Weeks from the Center for Biophotonics at the University of California, Davis.
I'm Thomas Huser from the Center for Biophotonics and the Department of Internal Medicine at the University of California, Davis.
Today we will show you a procedure for using doubly resonant coherent anti-stokes Raman scattering for producing chemically specific images of cells.
We use this procedure to detect signals from molecules that are too weak to be detected directly by coherent anti-stokes Raman spectroscopy alone.
So let's get started.
In order to generate CARS and DR-CARS signals simultaneously, three tunable and synchronized short pulse laser sources are required. To obtain these synchronized pulses, start with a single short pulsed laser with an output power of 10 watts, a fixed wavelength of 1064 nanometers, a fixed pulse length of 7 picoseconds, and a fixed repetition rate of 76 megahertz. Using a series of half-wave plates and polarizing beam splitter cubes, the beam is split into three parts. A half-wave plate combined with a polarizing beam splitter cube allows adjustment of the amount of power.